Hi, this is Don Schaffner, Distinguished Professor and Extension Specialist in the Department of Food Science in the School of Environmental and Biological Sciences at Rutgers University. Today, I'm happy to bring you my thoughts on microbiological sampling Part 1 as part of Contact's mini webinar series. When we think about microbiological sampling, the homogeneity of that microbial contamination matters. Another way of thinking about this is that a field of leafy greens is different than a tanker truck of milk or a combo bin of beef trim. To further elaborate on this point, I want to ask you the question, how risky are 1 million pathogenic E. coli? Well, the answer is that it depends. Let's imagine that we have 1 million pathogenic E. coli, and we can control how those organisms are distributed in samples of food. And so let's imagine those 1 million E. coli spread out over different numbers of servings. For example, we can take all 1 million pathogenic E. coli and we can put them into one serving of food. Well, in that case, there's only going to be one illness because there's only one contaminated serving of food. But we could also spread that contamination to 10 servings, 100 servings, 1,000 servings, and on up to 1 million servings. Because of the dose response relationship of humans to pathogenic E. coli, we estimate that if we were to take 1 million pathogenic E. coli and we were to spread those million E. coli over 1 million servings, we would expect to see about 2,500 illnesses. If we spread those pathogenic bacteria over 100,000 servings, we would see almost 25,000 illnesses. And if we were to decrease the number of servings further and increase the concentration in each of those servings so that we had 10,000 servings, we would see almost 1,500 illnesses. And again, you can see the other different assumptions in the graph on the slide. Now, let's imagine that we're trying to test those servings to find those pathogens. Well, this graph on the right-hand side indicates the fraction positive versus the probability of detection for an n equals 60 sampling plan for all of those different servings. And so where we took the 1 million pathogenic E. coli and we put them into 1 million servings, basically now every single serving of that food is positive for E. coli. So an n equals 60 sampling plan is, of course, going to find at least one positive because every single sample is positive. Now let's imagine instead of every serving being positive, 1 in 10 servings is positive. Well, chances are if we take an n equals 60 sampling plan and 1 in 10 servings is positive, it's almost inevitable that in those 60 samples, we're going to find at least one positive. If we change the fraction positive now to 1 in 100 servings, well, if we use an n equals 60 sampling plan where about 1 in 100 servings is positive, we're going to detect that about 50%, slightly less than 50% of the time. And again, if we decrease the fraction positive even further to 1 in 1,000, well, we are going to find that contamination some of the time, but as we drop the fraction positive further and further, we are less and less likely to detect the problem. So here we have a situation where if most of the servings are positive, we will find those positives most of the time, especially if we've got a very high level of positivity, like every sample being positive, or even every 1 in 10 being positive. And again, remember, if 1 in 100 are positive, we're going to still find that about half the time. So what does that mean? Well, that means that if we have that very uniform contamination, we are going to be able to prevent those 2,500 illnesses because we're going to detect it most of the time. And we're even going to be able to prevent those almost 2,500 illnesses that come when 1 in 10 samples is contaminated. If it's 1 in 100, we're only going to be able to prevent those 1,500 illnesses or slightly less than 1,500 illnesses about 50% of the time. And again, you can see for that one serving that's contaminated with 1 million pathogenic E. coli, well, we're just hardly ever going to find that. Even 10 contaminated servings, we're not going to find. 100 contaminated servings, very rare. And even if we have 1,000 contaminated servings out of 1 million servings, where we're going to still make 400 people sick if those servings get consumed, we still have a very low chance of finding that. Let's summarize all of this data in a table. Here you can see on the left-hand side of the table the number of contaminated servings going all the way from 1 up to 1 million. We can also see the corresponding number of cases going from 1 all the way up to slightly more than 2,500. And then finally, in the rightmost column, you can see the probability of detecting that level of contamination using an N equals 60 sampling plan. And again, I made some assumptions here that the sample size is equal to the serving size. I've also made the assumption that we can find as low a level as 1 E. coli per serving. But these are relatively reasonable assumptions, and they certainly help to make the point that I'm trying to make here. That point is that if you have a very high level of contamination, you have almost a virtual certainty. Even 1 in 10, you're still going to find that about 99% of the time. 1 in 100, you'll find that almost 50% of the time. But for those lower levels of contamination, which again still can result in a significant number of illnesses, you're very rarely going to find those problems. Let's summarize. The risk from 1 million E. coli cells is different depending upon those cells are spread amongst a different number of servings. A low level of E. coli uniformly spread amongst many servings, it's going to be relatively easy to detect. Whereas high levels of E. coli concentrated in a small number of servings are almost impossible to detect and may cause small or even relatively large outbreaks. Outbreaks with hundreds of cases caused by thousands of servings have a very low odds of being detected by an N equals 60 sampling plan. Thank you very much for listening. This has been my thoughts on microbiological sampling, part 1.